Hi, my name is Esther and I'm going to tell you about the Experimental Design Assistant, an interactive web-based tool to provide bespoke feedback on experimental plans for in vivo studies. So I work for the NC3Rs and I'm just going to give you a bit of a background about us as an organisation. So we are the National Centre for the Replacement, Refinement and Reduction of Animals in Research. We're an independent scientific organisation funded by the UK government. We were established in 2004. We lead on the discovery and application of new technologies and approaches to replace, reduce, and refine the use of animals in research, which is traditionally known as the 3Rs. And partnership is really key to everything that we do, and we work with scientists and organisations across the biosciences sectors, including universities, other funders, pharmaceutical, chemical, and consumer products industries, as well as regulators. Most of our budget goes towards funding research, and we are the main funder of 3Rs research in the UK. But we also have in-house programs led by scientists in the office. And one of these programs is the Experimental Design Program, which I work on. Our Experimental Design Program addresses concerns around the reproducibility of biomedical research, and these are concerns which have also been identified by others. So this slide shows a report from the Academy of Medical Sciences, and they did this in collaboration with the BBSRC, Medical Research Council, and the Wellcome Trust, and they looked at improving research practice, and they were looking at potential causes for reproducibility and reliability issues in biomedical research, and they identified 6 causes, and I'll just quickly go over them. One was omitting null results, so this is also known as publication bias, when scientists or journals decide not to publish studies unless the results are statistically significant. Data dredging, also known as p-hacking, so this involves repeatedly searching a data set or trying alternative analyses until a significant result is found. Weak experimental design, so a study may have one or more methodological flaws that mean it's unlikely to produce reliable or valid results. Underpowered studies, so statistical power is the probability that the analysis will detect an effect of a pre-specified size if that effect exists. And an underpowered study is too small to reliably indicate whether or not that effect exists. And then there are things like errors, technical errors that may result within a study, so misidentified reagents or computational errors, and then underspecified methods. So a study may be really robust, but if its methods are not shared with other scientists in enough detail, then others can't precisely replicate it, so that's underspecified. And the EDA, which I'll be introducing you to shortly, can help you avoid data dredging, can make your experimental design stronger, and help you avoid an underpowered study. So these 2 papers on this slide have also really highlighted reproducibility issues, and they seem to have given the reproducibility movement a lot of momentum. So these papers describe findings of in-house validation projects from Bayer and Amgen. So before embarking on new translational efforts, most pharmaceutical companies routinely try to reproduce in-house interesting data from the published scientific literature. And these validation studies can take 6 to 12 months. But the issue is that in the majority of cases, the in-house data was found to be not consistent with the published data. So these 2 papers showed that between 80 and 90% of the projects where they conducted in-house validation studies, the published findings couldn't be reproduced. And there are many reasons for which findings may not be reproduced, but things like poor experimental design or reporting that's not transparent or reporting that's not complete have been flagged as concerns. So we've conducted a survey looking at the quality of published animal research. So we looked at publicly funded animal research in the US and the UK and experiments using mice, rats, and non-human primates across all research areas. So this project was funded by the Medical Research Council and it was published in 2009. And so we identified key areas for improvement in experimental design analysis and reporting. So for example, very few papers reported the use of randomization and blinding, and none of the papers discussed how the sample size was chosen. When we looked at the statistical analysis reporting, we found only 70% of the papers described the methods used and presented the results with a measure of precision or variability, such as a standard deviation. So that means 1/3 of the studies were lacking the minimum information required to understand the results. And then looking at that, after looking at that, we looked at the type of information included in the papers. So a whole range of things like how the experiments were described, how the animals used were described. And we found, for example, that 1/4 of the studies reported neither the weight nor the age of the animals being used. So the survey really identified scope for improvement in the way animal research is conducted and reported. And these findings are not isolated, and virtually every study that's looked at the quality of animal research has found quality wanting, and there hasn't been a lot of improvement in the last 10 years. Though this study, published more recently from Malcolm McLeod's group in Edinburgh, was looking at highlighting how information allowing readers to assess how reliable the results are likely to be is frequently missing from publications. So it's looking at some of the same things that our previously published survey looked at. So in panel A, we have frequency of reporting randomization, panel B, blinded assessment of outcome, and panel C, sample size calculation. And this was done across different disease areas for in vivo research. And in the bottom right is a list of the disease areas, and on the graphs they go on the x-axis from left to right in that same order. And you can see that in panel A with randomization, although it did vary across disease areas, on average about a quarter of the papers reported randomization. And in panel B, although it did vary across disease areas again, just under a third on average reported blinding in the research publications. And you can see in panel C, sample size calculation, that is quite consistent across the board, and less than 1% of the papers studied showed any justification for the numbers of animal used. And you might think that this depends on where you publish, and that papers in high impact factor journals will publish more rigorous science, but the study also looked at this and they found that it doesn't make a difference. So if we look here, panel A again is looking at randomization, panel B at blinded assessment of outcome, and panel C sample size calculation. And as before, the percentage of papers reporting these are on the y-axis, but on the x-axis here we have deciles of journal impact factor. So it's increasing from left to right. And you can see that there's no correlation between the higher impact factor journals and higher reporting of measures of rigor. So this really highlights the problem with the reliability of animal research and whether we can actually trust the results that are being published in the literature. And this is also an ethical problem, because if studies are not designed to yield robust results, or if they're not reported properly, then it is a waste of animals. And it is an ethical issue, but it can also be an economic issue. So this paper was published a few years ago, and it looked at how much all of this irreproducible research was costing. So the budget for preclinical research is about $56 billion a year in the US. And they estimated in this study that about half of that was producing irreproducible research. And then they looked at within the irreproducible research, what were the categories of irreproducibility, and they found that about half of that was down to the study design, the analysis of the data, or the reporting. And those are things that our program of work at the NC3R specifically focuses on. So the experimental design program at the NC3Rs aims to help researchers improve the design of animal research and its reporting in scientific publications. So our perspective is that if studies are not designed to yield reliable results, or if they're not reported in enough detail, they can't add to the evidence base, and that means they're a waste of animals. So we've developed a number of resources to help support scientists in designing their experiments more robustly and reporting them more thoroughly. And today I'm going to talk to you about the Experimental Design Assistant. So the Experimental Design Assistant, or EDA, is an online tool for researchers to design in vivo experiments. So it's freely available, and it's available on eda.nc3rs.org.uk. And the EDA is like having your own personal statistical assistant. So it provides bespoke advice and feedback on your experimental plans. It gives you recommendations on statistical tests that are appropriate for the design of your study. And it helps determine the most appropriate sample size for your study and provide dedicated support for randomization and blinding. And you can produce a report to summarize the key information in your experimental design as a PDF. So in the EDA, each experimental plan is represented as a diagram made up of colored shapes called nodes, and each node represents a different part of your experiment. So on the slide here, we have a simple two-group comparison, and I'm just going to talk you through this diagram. So we start with a pool of animals, and the animals are allocated by complete randomization into two groups. Group 1 receives an injection of vehicle, and Group 2 receives an injection of a drug. And then we measure the plasma glucose in all of the animals in our experiment, and the data collected, so the plasma glucose levels, are used in the analysis, as well as the independent variable of interest, which in this case is whether the animals received a vehicle or drug. So an EDA diagram has three kind of main types of information in it. So in the gray nodes in the top left here, we have high-level information about the experiment and the objectives and things like that. In the blue and purple nodes here, we have the practical steps. So these are the things that you physically carry out in the lab. And in the kind of green and maroon color, we have the analysis plan. So things that are important when you're analyzing your data. So there is more information inside each of these shapes. So for example, in the group nodes, you'll have the number of animals per group, the number of experimental units per group, and justification for the sample size. And then in the allocation node, you'll have more information about the procedure for randomization. So let's have a look inside an allocation node. So we can see the randomization strategy. So you can select from your drop-down list. In this case, it's complete randomization. But you can also name the randomization procedure. So that's how the random sequence was generated. So in this case, a spreadsheet generated by the EDA. And you can see that there are other fields inside the property of this node. So you can put more information inside each of these shapes in the experiment. And an EDA diagram can be more explicit than a narrative text description of an experiment because you've got more information inside each of those components. And a good example of this is blinding. So often in a text description of an experiment, you'll say the researchers were blinded. But in an EDA diagram, you can say that the researchers, whether or not they were blinded at the allocation stage, when you're taking the measurements, and during the analysis, and not only whether or not they were blinded, but how that blinding was carried out. So for example, at the measurement stage, whether the animals were coded or whether somebody else took the measurements who's not involved in the experiment. So you can be really explicit about what's going on. And you can represent any experiment as a diagram in the EDA, and the diagrams are machine readable. So the system uses computer-based logical reasoning to provide feedback. So some examples of the feedback include this prompt, which says that the diagram does not include any nuisance variables. So the prompt includes what a nuisance variable is, common types of nuisance variables in animal experiments, such as baseline body weight or the day of the experiment if there's day-to-day variability, and also how to use that variable in the design and analysis of the experiment to try and mitigate against the effects. Another feedback prompt asks you to clarify the blinding status when the results are assessed. The prompt will explain why the blinding is important, different stages of the experiment during which you can be blinded, and different ways to blind different stages of the experiment. So you can decide not only what is most appropriate for your experiment, but what is actually feasible in the facility or the laboratory that you're working. So you can also use the EDA to help communicate your plans with others, such as an ethical review board or a funding body. So the EDA report was designed to help researchers who are already entering this information into the EDA to export key information at the click of a button and use it to identify this key information in the grant application or ethical review process. So the report contains the objectives and hypotheses of your experiment, the number of animals you're using and the justification for the sample size, any steps you're taking to minimize bias, such as randomization and blinding, what your primary outcome is and secondary outcomes if you're measuring more than one outcome, and your planned statistical analysis methods. And then there's a summary of the EDA feedback that has not been addressed and an image of your EDA diagram at the end. So we've come up with this EDA workflow to help you get the most out of the support we provide in the EDA. So what we suggest you do is you draw your diagram and then you fill in the information inside each of the nodes. So fill in the node properties, but if you're not sure what to put in any of those fields, then we suggest that you leave it blank because the EDA can provide feedback to help you decide what's most appropriate. So once you've had a first go at drawing your diagram, you can use the critique function, and then it will give you some feedback, and you may go back and decide to redraw parts of your diagram, change it a bit, add more information in the node properties now that you've read the feedback, and then you can critique your diagram again. So you can repeat these first 3 steps as many times as you like until you're happy with your experimental plan. Then you can use the analysis suggestion function, and that will recommend an analysis method that is appropriate for the particular design that you've put in the EDA. And once you've done that, you can use the inbuilt power calculators to determine the most appropriate sample size. You can use the randomized allocation report function to generate your randomization sequence. And what this does is it generates a randomization sequence taking into account any blocking factors, and it generates that as an Excel spreadsheet attached to an email. So the system will ask you to provide the email address of the person who's helping you with randomization and blinding. So the random allocation sequence gets sent directly to them so you can maintain your blinding throughout the experiment. You can also share the experiment with others, so share your diagram with colleagues and collaborators. And you can produce this diagram report, the PDF we talked about in the previous slide. And once you've carried out your experiment, you can go ahead and update the diagram. So there are a couple of fields that are specifically for once your experiment has been finished. So for example, in the group node, there is a space to say the planned number of experimental units, but also the actual number that you've used. So if you had some attrition during the course of your experiment, you can record that. And that's really good because you can hang on to that record of attrition. And then if you come to do a similar study in the future, you can look back and you can go, right, okay, I had 10% attrition. So you'll take that into account in your next sample size calculation. There's also more information on experimental design on the EDA website. So this covers concepts crucial to the internal validity of your experiment and how best to represent these in your EDA diagram. So an example of this is in our animal characteristics page, where we provide guidance on how differences in animal characteristics can influence the experimental design used. So an example of this guidance is regarding using both sexes in experiments. So using both male and female animals in an experiment enables the findings to be generalized across the whole population, so you're not just restricted to one sex. But one of the reasons researchers don't always do this is that they believe that the variation in female animals, especially across the estrus cycle, can introduce extra variability. So this graph on this slide is plotting the coefficient of variation for different experiments from a meta-analysis study. So this study looked at 293 articles with behavioral, morphological, physiological, and molecular traits that were monitored in male and female mice. And these female mice were tested without regards to the estrus cycle, so they didn't control for that. And you can see that by plotting the variability, so towards the right-hand side of the x-axis means more variability, and the data from the females is plotted in red, and the data from the males is plotted in blue, and where they overlap, it is purple. And you can see that the variability was not greater in females than in males, so that's not a good reason to not include females in your study. So this graph is from a different study looking at sex bias, and this time they were looking at sex bias in pain research. So the graph shows analysis of preclinical studies published in the journal P.A.C.E. So the bar on the left covers research published between 1996 and 2005. And it shows that about 80% of the studies in this period used only males. And then the other bars are 2015, 16, 17, 18, and 19 across from left to right. And it shows that there is, over time, the male-only studies have decreased as more studies are conducted in either females or in both sexes. And the studies including both sexes are shown in this graph in the 3 shades of purple. So these different shades represent how the sex data were analyzed. So the darker purple, where both sexes were included but the sex differences were not analyzed. And then the other 2 lighter shades of purple indicate where sex differences were looked for but either not identified in the mid shade of purple, or a difference was reported in the lightest shade of purple. So you can see that we are making progress towards using both sexes in our studies, but there is still some more work to be done. So it does make sense to include both sexes in your study unless you are studying a process like oogenesis that does occur only in one sex. So using both sexes doesn't necessarily mean you need to increase the total number of animals in your experiment. But to ensure that you have the same proportion of males to females in each of your experimental groups, you should randomize males and females separately to your different intervention groups. And this prevents sex bias in those groups. If you are using both sexes to make your results more generalizable, so you want them to be generalizable across the whole population, but you have good reason to believe that you're not expecting a big difference between males and females, then you don't need to increase the total number of animals you're using, and you can determine the sample size by powering for the biologically relevant effect size of your main intervention, say your drug treatment, and use both sexes with preferably half and half male and female in each group. And if you include sex in the analysis as a blocking factor, then this takes into account the variability induced by sex and increases your ability to detect a real effect of that intervention. If you do want to compare male and female animals to see if the intervention affects them differently, you would design your study with 2 factors of interest, so the intervention and sex. And the number of animals you need in your study depends on the purpose and what comparisons and which groups you're going to be comparing afterwards. And so we do have some guidance on this on our EDA website in the experimental design section and the animal characteristics page. So why should you use the EDA? We believe that using the EDA improves the reliability of your results. So it helps you address obvious sources of bias, and this helps you address them while you're planning your study. So that means that if you do find a potential source of bias, you can do something about it and mitigate against it so that you can fix it before you've started your study, so it's not too late. It can also help promote better understanding of experimental design, so raise awareness of why these things are important. So every time a user gets feedback from the EDA, you learn something new. And the system is not a black box telling you exactly what to do, but it does provide information about the implications of doing things a certain way, so you can understand what you're doing and make informed choices. The EDA can also facilitate peer review or assessment of your experimental plans. Because it's such an explicit description, then it can be really transparent. So editors, peer reviewers, and readers having access to your design and all the information have a really good idea of knowing exactly what you're doing. And also it can form a sort of soft pre-registration. So it can provide evidence that your study was planned as you're reporting it and that you haven't, for example, changed your primary outcome measure after looking at the data. And this is because the EDA report, the PDF report I talked about earlier, when you create that, it has a timestamp on it. And if you download that report and then maybe put it on something like Open Science Framework or another data repository, you'd also have an upload timestamp for that as well. So you could have two kind of ways to prove that you have planned your study as you are reporting it. The EDA can also promote more careful consideration of your experimental plans. So we'd really want scientists to spend more time planning their experiments, identifying their variables, considering the data you're going to be collecting. So we want scientists to spend more time planning and thinking about things before they've started the study. So if you discover potential problems, you can mitigate against them or you can fix them, rather than completing the study, realizing afterwards that you've introduced biases, but there's nothing you can do about it. And we also think the diagrams are really useful to help discuss your experimental plans in lab meetings or workshops, because having a visual overview of your experiment makes it easier to discuss the specific thing you want help with, like, for example, figuring out how people are going to help you with the blinding or discussing which is the best primary outcome measure for your specific scientific question. So the EDA helps you design your experiments, but if you want guidance when planning how to breed your experimental animals, then the NC3Rs has breeding and colony management guidance online as well. So the guidance has been produced by an international working group of experts in colony management and experimental design, including experts from the Jackson Lab in the US, and the Mary Lyon in the UK, and the Centre for Phenogenomics in Canada. So the information is available as a web resource, and it includes best practice for managing your colony, considerations when archiving your lines, and tips for selecting a breeding strategy. For example, the things you need to watch out for that may be affected by the genotype or the time frame when your animals are needed. Do you need a certain number of animals every 6 months, for example, or do you need a whole lot of animals all in one go? It also includes a frequently asked questions page, which includes a worked example to calculate breeding numbers. We also have an email advice service for very specific questions. For example, if you're having trouble breeding a difficult line, then you can email us and we can forward that on to the expert working group to get some help. We are having a webinar on efficient breeding on the 1st of March 2021. And if you are interested in joining us, you can go to nc3rs.org.uk/webinars and you'll find a way to register there. So the EDA helps you plan experiments, and the breeding and colony management guidance helps you plan the breeding of your experimental cohorts. But we developed the ARRIVE guidelines to improve the reporting of information in animal research publications. So the ARRIVE guidelines were originally published in 2010, and they've been revised over the last few years, and the updated guidelines, ARRIVE 2.0, were released in July last year. So the guidelines consist of a checklist of items that have to be included in a manuscript to ensure comprehensive and transparent reporting. So the items in the ARRIVE guidelines are important for different reasons. So for internal validity, for example, description of study design, how the animals were allocated to groups, how the sample size was chosen. So this is really crucial information to understand how reliable and robust those results are. Some of the items are important to determine that, to ensure the paper has enough information to repeat the studies that are described. So a comprehensive description of experimental procedures and description of the characteristics of the animals in the study. And then some items provide information about the context and scientific relevance of the study. So for example, the scientific background, any relevance to other species, and information about the ethical approvals. So the ARRIVE guidelines apply to all in vivo research. So it doesn't matter if the species used does not require ethical approval. The guidelines are about rigorous science, so that applies to all in vivo experiments. In ARRIVE 2.0, the items are organized into two sets. So the ARRIVE Essential 10 is the minimum requirement. So this means the basic minimum to include in a manuscript, because without this information, reviewers and readers can't confidently assess the reliability of the findings presented. And then there's a recommended set. So these items complement the Essential 10 and add really important context to the study. And reporting all items in both lists represents best practice. And we have introduced a couple of new items. So for example, protocol registration. So pre-registration is now standard practice for clinical trials, but it's also emerging as a mechanism that is likely to improve the transparency of animal research. And for that reason, protocol registration is one of the new items that we added to the newly revised ARRIVE guidelines. So protocols can be registered in a registry, but another option is via registered reports. So registered reports is a publishing format where you have peer review of the study design, including your analysis plan, before you start your study. So this means that reviewers' comments and suggestions can be taken into account before you begin the practical experiments, and this can really help you make your research stronger. So after this initial review, called stage 1 review, you get in-principle acceptance of your study. So this means you can publish your study regardless of whether your results are statistically significant or not. So this really encourages more rigorous study design and helps reduce publication bias. And the registered reports format is used by over 250 journals, including BMC Biology, Royal Society Open Science, and PLOS Biology. And I've put a link to the registered report website on this page, so if you'd like to learn more about them, you can check them out there. Another important initiative I'd like to highlight is Reproducibility. So Reproducibility is a grassroots initiative helping researchers discuss issues, papers, and ideas about improving science, reproducibility, and the open science movement. So it's open to researchers in any field and at any career stage, so anyone can get involved and anyone is welcome to start their own reproducibility club. So if you're interested in finding out more, their website is reproducibility.org, and they have starter packs on the open science framework, and I've put a link on the slide to their open science framework repository. So the resources on OSF include reading lists to help you get started, and there is one tailored specifically towards in vivo studies. So I'd just like to conclude by highlighting the reach of ARRIVE and EDA, and so they are endorsed by many of the major stakeholders in scientific research. So for example, the ARRIVE guidelines are endorsed by over 1,000 journals, so they're recommended in their instructions to authors, and many major funding organizations recommend that researchers use the EDA when preparing a grant application that contains animal research. So that includes the MRC and the Wellcome Trust in the UK, but also funders in Europe or the US, like the NIH. And there's also grassroots support from the researchers themselves. So the EDA has over 10,000 registered users, and over the past few years, we have had over 40,000 requests for printed copies of the ARRIVE guidelines from 50 different countries. So thank you very much for your attention. If you'd like more information about the EDA, you can go to eda.nc3rs.org.uk. And if you'd like more information about the ARRIVE guidelines, the website is arriveguidelines.org. Thank you.